So my name is Antonio Giraldez, and it's really a pleasure to be here, Thomas. My story with MBL, so basically I first heard from EMBL from some of my colleagues in the south of Spain. And when I was a student in Madrid, I basically wrote point blank to Steve Cohen and say, well, I want to do an internship in the summer. And after being internship here, without barely speaking any English and hardly understanding Steve Cohen, it was still a wonderful experience. And then decided to come back for the PhD. And I think coming in as a PhD student was really an incredible and unforgettable experience that has marked me for many years. In particular, the international aspects of EMBL has really left a stamp on me that now I try to continue in my own laboratory and replicate that amazing community that we have had here of students, where I think in my lab we have at the moment about 16 nationalities, which is kind of a little bit of a mini replica of the EMBL spirit, and also the collaborative spirit, which has been quite amazing.
So I know you came with already some training with a model system. You got involved in the same model system, but tell us a little bit of what you got involved in when you joined Steve Cohen's lab, and what was the science back then?
So when I got involved with Steve Cohen, we were really trying to understand the signaling molecules and that pattern morphogenesis in organisms, and in particular during development of the Drosophila. So we and it was already ongoing, started a screening that you were also part of, large screening in Drosophila, and in particular I was interested in the patterning molecules of the wing. So I remember really fondly the first year was just screening, like looking at a window out of the flight room, looking at new patterns. We found over 200 different genes that affected the morphogenesis of the wing. And it was just every day was discovering new genes that had an effect on development, and that was just an incredible experience. And that led to the discovery of some interesting genes that are regulators of wing signaling, but it was really just pure discovery and joy and a lot of hard work, but it was so much fun.
So I remember that time and the passion that we had, and maybe one question for you is a reflection and a few words on the role of these approaches that are very bottom up. Somebody would call them fishing expeditions, but then of course they are the basis for knowledge. So it's something, how do you see it? I mean, should we put some money in fishing expeditions? Should we be more top down on specific hypotheses?
Yeah, so I think that this kind of fishing expeditions or genetic screenings, they really allow you to discover unexpected things. I think that when we have screenings where you basically do kind of a prediction of top-down, I think to some extent it can limit the extent of the potential for discovery. And I think in these genetic screenings where basically the biology will tell us, what affects, in this case, the morphogenesis of the wing or genesis or planar cell polarity. I think in that case, it was really the fly was telling us, well, this gene doesn't allow the fly to make wings, so it's basically an inhibitor of wing development and so on and so forth. So I think that, in fact, that led me to a gene that was previously undiscovered and uncharacterized. So I think that the power of discovery from these screens is really important, benefits.
I mean, one thing that I remember, and you have been, of course, more part of it than I have been, was that at the time, with all these screens, one of the things that emerged was actually all the regulation that is the microRNA regulation, right? That nobody would expect, because we were thinking in terms of protein encoding genes.
Yeah.
But this is, I guess, one. You could think it was actually, if you remember David Hipner, it was actually some sort of a byproduct. It was that allele that you could map because there wasn't a gene.
That's right. That's
so
right. No, that was a really interesting discovery by a group of students and postdocs that at the end it got cloned by Julius Brennecke and initially characterized by David Hipner, where again something that was completely unexpected, where a microRNA was when overexpressed was really causing a growth effect, and that led to one of the first microRNAs in Drosophila. And actually, for me, when I left EMBL, became a little bit the inspiration to then try to understand microRNAs in a vertebrate system that formed the basis for my career then in New York and then at Yale. So I think that's also a beautiful example of how this kind of genetic screenings, in this case, was overexpression of different genes randomly based on landing of a P element, I think can lead to really unexpected discoveries.
The other aspect that you just mentioned that I find wonderful of the scientific community is how you can gain and take advantage of somebody else's knowledge and then try to decide to apply it in an entirely different system to just go and build new knowledge. So how did your science develop then after the MBL? You mentioned that you went to a postdoc, and then of course you got your current position at Yale. So
it developed. It developed really serendipitously. I think part of it was the inspiration from Steve Cohen and his teachings on science, and Julius Brennecke on the microRNA. Part of it was really the advantage of having a mentor like Alex Shear that really provided a tremendous amount of freedom in pursuing the research topics, related to development, but really unconstrained. And I think the passion and the curiosity to try to understand what these new microRNAs might be doing in invertebrate development. I think that's how I got into the projects that I was doing as a postdoc. Basically, the first steps were really, are there microRNAs in zebrafish, and then trying to understand and pulling the thread little by little, and then led us to the discovery of how microRNAs shape gene expression during the maternal to zygotic transition as a way to the embryo kind of forgetting the initial instructions that the mom has given so that it can start its own development. So that was a really interesting paradigm that had been observed for many years, but we didn't know the molecular factors that were doing it.
So let me ask you maybe more of a personal question, because I know you a bit from that time. So one thing that always amazed me of your approach to science is the incredible motivation, passion, and drive that you had. I remember at some point you had to do a critical number of experiments very fast, so you said, I'm just gonna close myself in that little room and just do the science out of that. So maybe you can share, do you have a recipe for just getting that level of motivation? Because science most of the time is frustration. So a lot of people actually get out of this because they, most of the experiments don't work, and they say, why, I cannot really take it anymore. So how is it that you were able to be so driven?
I think several ingredients. I think the one factor that really drove me was my obsessive compulsive nature, actually. So I think that, like the fact that when I'm doing something that I have fun, I just, I think people talk about the work-life balance, and I think when science is your life and is your passion, it's like
there is the balance.
There is a perfect balance because you're just spending all the time playing. The other thing is that I remember the last year of my PhD was very frustrating because we were trying to understand the mechanism, and I felt like just walking and banging my head against the wall. But I think that, there are some moments where things work, and that kind of provides a renewed fuel for that passion that I think allows you to keep going. I think so. It's that combination of, having amazing mentors that really support you, but also this obsession when I'm doing something that I like, and the few moments dispersed that provide that extra fuel that allows the passion to keep going.
Yeah. So I can certainly relate. It's that you have to really be able to enjoy that kick when
it comes.
When it comes and things,
which is seldom at times. Yeah.
But then another aspect that maybe I want to ask you about is that connected to that there is always the competitions, right? Many labs that are trying to do similar things, and sometimes a system of publishing that only rewards the first one. So maybe do you have some thoughts about whether this is, the way to go, or we should allow to have a more relaxed publishing world to be able to capture more of that? So what is your ideas after years of career now?
Yeah, so I think that sometimes, at least to me, so publishing is a double-edged sword. In one way, publishing is a wonderful way to make science known to your colleagues and advance science around the world. On the flip side, I think publications can be a detrimental aspect of science from the point that it really can slow down science in the way that peer review process has evolved to unreasonable levels of requests. So I think that, bioRxiv, I think, provides a wonderful system where you can make your science, available to everyone immediately. And I think, to me, has been one of the most satisfactory elements, where it's instant gratification, where you can put things in bioRxiv and get feedback from your colleagues. I think the other element is the competition, and I think that I over the years, even though I was very competitive, and I remember when I think the first of our colleague, Anton Enright, had like a Nature paper within the first year. So I think there are two levels of, one is the peer pressure and the other is the self-imposed pressure. So that was really a remarkable moment. And that also is can be a little bit of a motivation, no? To not as a competition, but as a motivation from your colleagues when they are doing well. But I think I take an approach more of a collaborative approach, where I think that, I think being the first, if you're naturally, have discovered something that is, quite remarkable and unique, and you are the first, that's wonderful. But I think I take an approach where if that comes, that's great. I think it's more gratifying to collaborate with others to bring complementary expertise, which is also an amazing theme at Yale, where you can benefit from the mutual collaboration across disciplines to really be in an area where you are the first one exploring that based on the possibilities of collaboration with multiple techniques of expertise. And I think that's a way that I think is much more rewarding than just, have a group of people going on a race looking at the same aspect, no?
Yeah, no, I think I can see that. One way perhaps to do that is creating an environment that is so, forefront, so that you naturally will be among the first, but is a sort of collective endeavor, because it's part of the culture.
And I think that to be in the forefront, I think one of the recipes, I think, is to really combine techniques or combine approaches in a way that is that interface of collaboration that really creates the possibilities of discovery that are not possible by other individual laboratories, which I think allows that competitive edge.
So maybe, going from there to the fact that you have been, director of your department, right?
Yeah.
So you becoming more and more involved, I guess, in systemic approaches to science. I mean, what do you find challenging of that aspect? I mean, in a way, it's a new job, right?
Yeah, so I've been chair of the Department of Genetics for 7 years at Yale and just stepped down in June. And what I found most, before the challenge, what I found most rewarding was actually to be able to support the science in the department with the resources that were available to us, but also to recruit amazing talent, So I think that's another area where I felt really excited and motivated, where we were able to shape the department through the recruitment of amazing scientists that complemented many of the incredible scientists that were in the department. And we were able to recruit scientists from embryoids, human embryoids, to ancient DNA, to physicists looking at gene expression through and genome conformation to human genetics. So I think that shaping and providing the resources to the new wave of scientists for the department has been really the most exciting. I think the challenges are sometimes the management of the organization. I think managing, when you have a large organization that has many different demands, where maybe science, top quality of science is just one of the pressures, that I think has been one of the challenges, and also a great learning opportunity.
Okay. Did you have a chance to apply some of the lessons learned at the EMBL? I know you've been a student at the EMBL, maybe you were not so involved in this sort of perspective, but were there something that you found useful of the time you were at the EMBL?
I think so. I think for me, like the teachings of the EMBL have come with me and are very dear to my heart. And I think I have applied not just in my laboratory, but also in the way we recruited PIs across a large diversity of backgrounds, ethnicity, and topics, with the idea of creating that milieu of amazing science and amazing people that really can forge wonderful collaborations and advance science in ways that it wouldn't be possible. So I think that has been, to me, a teaching that has come with me and that I have been able to implement not only in my lab, but also in the department. And I think, the other teaching, I think, from EMBL is the mechanistic understanding of nature, which, is really at the source of the molecular understanding of biology that has also come with me, and try to apply that to the way I organize the department and the recruitments, So I think that's exciting, and now I feel, I have one of the best environments at Yale to do science across multiple disciplines.
So maybe another questions more related to your work, or more related to the system that you have been using, because you have been using zebrafish for a long time, and before you were a Drosophilist, is how do you see now the role of animal models in a system and in an ecosystem that now seems to think that we can do things with cells and reprogrammed cells and pieces of organism and organoids? I mean, do you still think that there is a role and some need to found fundamental research with these type of models?
Yeah, I think that the model systems really provide an incredible tool of in vivo analysis of the biology that we are studying, which I think is also complementary to other tools that are available, such as cell culture, organoids. I think that, as the same way that we can think about collaboration between different techniques, different laboratories, we can also think about the collaboration of different systems, from organoids to animal systems to cell culture. I think that what the new era of genomics allows us now is to basically, and genome engineering allows us to use basically models that were not the classical model systems that are now available for research, that I think open the possibilities, as we have seen with the TARA project, to learn about the incredible diversity that there is around in the world, and the limited window that we focus sometimes on. So I think that is really exciting, and I think that part of the future will be to dig through the hundreds of thousands of genes that have been discovered in this new species, which I think is really exciting.
Perhaps, coming back to the network of EMBL and all the connection that the EMBL has provided, I mean, is still a network that you use nowadays, I mean, for the science and for your social life? I mean, are you still in contact with a lot of people in EMBL?
Yeah, no, I think that EMBL really comes with you over the years, and I think we have now colleagues across many universities around the world, and also across academic and non-academic areas where it's typical that I will go to an institute in a particular city and found a former EMBL colleague, let it be Vienna or Canada or Italy. EMBL to me means an amazing science with an international community that has basically created the foundation of my education, but also has allowed me and many other scientists to really empower us to make exciting discoveries that hopefully will come back and help humanity.